Hello, my name is Jacob O'Connor. I'm a fifth-year graduate student in the Baker Lab, and I use they or he pronouns. I'm a member of the BPSD program, which is an umbrella program, and because I'm in David's lab, that means I'm in the biochemistry department, and I'm getting my PhD in biochemistry. I grew up in Yakima, Washington. It's this small city in eastern central Washington, very different than Seattle. It's very arid. It's a very rural farm community. And growing up, I never really thought of science as a career. To me, scientists were Miss Frizzle and Bill Nye. And it wasn't until I got this opportunity in high school to come visit the Hutch Cancer Research Center here in Seattle that I first realized, whoa, like scientists are people, and I could be a scientist one day. Also, that visit kind of awakened in me a real love for structural biology. So during that visit, they gave us a presentation on sickle cell anemia, and they showed the two crystal structures of hemoglobin in its native state, and then the mutant version responsible for sickle cell. And I have sickle cell trait, so I have one copy of the mutant version. So I don't have any of the symptoms, but I've always been very aware of how hemoglobin influences my biology. And getting to see with my own eyes how this one tiny little single amino acid mutation results in a noticeable change in the structure that you can see with your eye on a crystal structure, and then how that structural change results in aggregation, which results in health problems, was just, it was mind-blowing. I was like, whoa, this is so cool. And ever since then, I've been really interested in thinking about the chemical and physical principles that underlie biological systems and how we can test them and learn more about them and use them to our own advantage. So after high school, I went to undergrad at Whitman College in Walla Walla, Washington, a small liberal arts school in a small rural town in eastern Washington. And I had a great time. I really enjoyed my time there. And as part of my time there, I did research with one of the professors, Dalia Biswas. And her lab focuses on using QM computational modeling to study enzyme active sites and also to build organic molecules that mimic those active sites based on those models. So in her lab, I really learned that I enjoyed the process of building a computer model in the dry lab and then going to the wet lab and running an experiment based on that model and then going back to update the model in the dry lab in the kind of iterative process. So when it came time to graduate, I was thinking, well, I think I want to be a professor. I really like science. I like teaching. I like doing research. Professor sounds great. That means grad school. What do I want to study? What do I want my grad school career to be about? And I was like, well, I like this computational kind of approach. I really like structure, function, relationships. Protein design sounds so cool. I'd love to do protein design. So I looked at a bunch of grad schools, applied to places that had professors working in that area, got accepted to a bunch of places, and chose here. UW is obviously a really good place for protein design. We have the Institute for Protein Design. It also was just a good culture fit, right? I'm from the Pacific Northwest. I kind of like the Pacific Northwest way of doing things. People felt I enjoyed my time here, and I felt like I would be happy working with the people here for the next 5 years. So I came, and now I research how to design very, very small proteins in the range of 6 to 12 amino acids, these tiny, tiny little peptides that have some sort of chemical constraint that makes them cyclic, so they're macrocycles. And I study how we can use design to try to make them better drugs. So on the one hand, I'm focusing on, or I'm looking at how we can design them in such a way that they passively cross cellular membranes so that they can be used, go after intracellular targets and be taken orally rather than injected. I'm also studying how we can use our computational methods to better incorporate non-canonical amino acids, because these peptides are chemically synthesized, and so we don't have to stay in the normal realm of 20 amino acids. We can do all sorts of cool, crazy, interesting chemistries. So using a computer to do that more smartly would be useful. Beyond research, one of the things I really like about UW is that I've had time here to grow in other ways beyond just as a research scientist. And I think that's really important. I think grad school is structured in such a way that it really motivates and incentivizes you to just be a research scientist, but it's important to nurture those other parts of yourself. So I have a lot of friends and family in the area, right? I play a lot of video games and role-playing games with my friends. I watch horror films. I go outside and do hikes, and I like to swim. So like, there's lots of ways I spend my time not in the lab. But beyond that, in my professional identity, I've also kind of grown and changed. I came in thinking academia was like the best thing in the world. And being here has changed my mind on that. For me, something that's been really important is we have a labor union here on campus. So all of the graduate students, all of the postdocs, and all of the, and a subset of the undergraduate researchers are part of this group of people that negotiate contracts every 3 years with the university, setting base pay scales, healthcare benefits, protection against harassment, all sorts of things. And when we negotiated our contract last time, I became really involved and learned how I can like organize with my peers and change these systems, and also how these systems are broken in some ways, right? I learned about how the academy historically has been built for one type of person, the straight cis white male from Europe. Like that's what the academy was built around, and that's basically who it's constructed to support. And for anyone who doesn't fit that, it can be really hard to get in and to flourish in this environment, which sucks. We need to change that. So now I don't think I want to be a professor when I graduate. I'm actually interested in getting involved with science policy. So that's looking at how politicians and rule makers use science to create regulations and laws, and also how those regulations and laws interact with how we do science. And that journey has been pretty long, and I've had a lot of help along the way. So I became very involved with science communication. I volunteered at the local farmers market, at the local science museum, to explain my research to a general audience. I've also worked with other graduate students to create workshops on how to communicate better. And I've been involved with our union and with graduate student policy groups here on campus. I've had a lot of different ways that I've been able to flourish these other parts of my identity besides just scientist. And I've also done a great deal of research and learned a whole lot there too, undeniably. I am getting a PhD in biochemistry after all. But at UW, I've had the freedom in the community to support all parts of myself and not just the box of research scientist, which I've really appreciated.